Welcome, dear students. Today we are going to discuss an important topic on classification of hormones. The core objectives of today's lecture are to know about the hormones in detail, to understand different mechanisms that cause a variety of effects in animals, to discuss various classes of hormones, and lastly, to know about the therapeutic uses of various hormones. To begin with, let us first discuss about the hormones. Years ago, Starling coined the term hormone to describe secretin, a substance secreted by the small intestine into the bloodstream to stimulate pancreatic secretion. So, the first hormone to be discovered was secretin. It was Starling who considered the endocrine and nervous system as two distinct mechanisms for coordination and control of organ function. Over several decades, a lot of exertion by biochemists, physiologists, and clinical investigators led to the characterization of many hormones secreted into the bloodstream from distinct glands and many organs. So, hormones can be defined as chemical messengers secreted into the bloodstream that circulate to the distantly located target tissues or organs in a well-regulated mood. Hormones are non-nutrient, low molecular weight, intercellular messengers effective in very low concentrations, usually in picograms, nanograms, or microgram. Hormones do not provide energy or building materials, but they do have effects on growth, differentiation, and metabolic activities of their target cells. They serve to coordinate the metabolic activities of several tissues or organs that accelerate or inhibit specific physiological process, and they do not initiate a reaction but can influence its rate of action. They are produced in an inactive form as preprohormones or prohormones, which are later converted into active hormone molecules. Hormonal regulation is achieved by the ability of hormones to have specific biological activity at their target tissues. Hormonal signaling, which represents a special case of communication between the cells in the body. Such signaling is important for energy production, metabolism, somatic growth, development, reproduction, and the ability of body to respond to internal and external stimuli. These complex interactions utilize controlled mechanisms of hormone synthesis and secretion and communication with other signaling molecules through various signal transduction pathways. Hormones are the characteristic of endocrine system, a complex interrelated mechanism of communication between the cells of an organism. Endocrine glands are the glands of an endocrine system that secretes informational messengers or hormones into the blood circulation, reaching to the target organ. Because such glands are not connected by any duct, therefore they are also called as ductless glands. The system is comprised of a diverse series of signaling mechanisms required for the regulation process and function vital for multiple organs. Hormones are involved in maintaining homeostasis, that is the consistency of an internal environment maintained for the benefit of a whole organism, as shown in the figure. Homeostasis was first recognized by Claude Bernard in the 19th century, who noted that the internal environment, that is the fluid bathing cells, had to be regulated independently of the external environment. Being able to regulate and maintain the internal environment, the animals are free from change in the external environment, allowing them to live in the changing or harsh environments. Though there are metabolic costs associated with maintaining homeostasis, homeostasis is maintained by negative feedback regulation. For example, an endocrine tissue produces a hormone that affects the production of a metabolite by the target tissue. The metabolite then interacts with the endocrine gland to reduce the production of that particular hormone. This forms a cyclic system in which the levels of the metabolite are maintained at a particular level. The set point of the system can be altered or changed to affect the levels of the metabolite by altering the sensitivity of the target tissue to the hormone or the sensitivity of the endocrine gland to negative feedback from the metabolite, as is shown in figure. In addition to maintaining homeostasis, hormones can also be used to drive change in an organism. In this case, levels of hormone increase to some peak, and this occurs by positive feedback. Positive feedback amplifies the response, so the tissue must be desensitized or turned over to stop the response. An example of this response is the surge of luteinizing hormone that leads to the ovulation, as shown in the figure. Luteinizing hormone produced by the pituitary gland stimulates the developing ovarian follicle to produce estrogen, which stimulates the hypothalamus to produce gonadotropin releasing hormone and increase luteinizing hormone production by the pituitary gland. This produces a surge of LH, which decreases only after the follicle ovulates. Hormones are cleared from the blood circulation by metabolic destruction, binding with the tissues, excretion by the liver, and excretion by the kidneys via urine. Overdose or deficiency of hormones will lead to several disorders in an organism, as is shown in the figure. Dear students, let's now discuss the general mechanism of hormones which cause a variety of effects in animals. Hormones can cause morphological change in organisms, such as differences in body shape and pattern between males and females. Hormones can act as mitogens to accelerate cell division or alter gene expression to trigger differentiation of cells. These stimulate the overall rate of protein synthesis or the synthesis of specific proteins. These are involved in stimulating smooth muscle contraction. For example, oxytocin stimulates contraction of myoepithelium in the mammary glands for milk ejection. Hormones affect exocrine secretions. For example, secretin, a peptide hormone from intestinal mucosa, stimulates pancreatic secretions, control endocrine secretions, and a number of tropic hormones from the anterior pituitary gland can stimulate or inhibit hormone secretion from the target organs. Hormones do regulate ion movements across the membrane and control permeability to water. For example, antidiuretic hormone or ADH or vesiprocin increases water reabsorption by the kidney, and hormones have a dramatic effect on behavior such as sex-related behavioral characteristics, maternal behavior, nesting activity, and broodiness. Dear students, up till now we have discussed about the hormones in detail. Moving ahead, let us now discuss the classification of hormones. Since hormones are very diverse, they have been classified into a variety of categories, like on the basis of chemical nature, mechanism of their action, and cellular receptors on which they act. Dear students, first let us discuss the classification on the basis of chemical nature. Hormones are classified into three main groups, such as proteins and polypeptide hormones. These hormones are defined by a wide range of size, composition, number of chains, modification of groups, and mechanism of production. Some examples are large single chain peptides such as the 192 amino acid growth hormone, the cyclic peptide of TRH that is comprised of just 3 amino acids, and prolactin. The anterior pituitary hormones, thyrotropin, thyroid stimulating hormone, follicle stimulating hormone, and luteinizing hormone are glycosylated and consists of 2 chains each, one of which is common to all 3 hormones, that is alpha chain, while the other is distinct and confers specificity to the hormones, that is the beta chain. Insulin is comprised of two chains that are derived from post-translational modification or cleavage of a single gene product, that is preproinsulin, while as adrenocorticotropic hormone or ACTH, melanocyte stimulating hormone or MSH, and beta endorphin are single chain proteolytic products of a large precursor molecule pro-opiomelanocortin. This group includes hormones secreted by the anterior and posterior pituitary gland in the body. Peptide hormones are the most prevalent and diverse among all. Now let us discuss the steroid hormones. These are derivatives of cholesterol containing a similar core known as the cyclopentanoperhydrophenanthrin ring. The synthesis of steroid hormones occurs as a result of enzymatically induced change to the cholesterol core. Synthesis of the adrenal and the sex steroids occurs in the adrenal cortex, that is cortisol and aldosterone hormones, and testes, that is testosterone, or ovaries, that is estrogen and progesterone. Such hormones are lipids or lipophilic derived from cholesterol as shown in table. Now let us discuss the third category, that is amino acid derived hormones. These are water soluble and are derived from amino acids. Specifically, the amines are derived from the amino acids tyrosine and secreted from the thyroid. These are termed iodothyronines and hormones from adrenal medulla. The precursor of the iodothyronines is thyroglobulin, a glycoprotein that is synthesized by the thyroid follicular cells containing tyrosine residues greater than 100. The iodothyronines are formed by iodination and coupling of two tyrosines and are the only iodinated compounds with significant biological activity. In the adrenal catecholamine secreting cells, tyrosine is converted sequentially to dopamine, norepinephrine, and epinephrine. The hormone serotonin, that is the 5-hydroxytryptamine, is derived from the amino acid tryptophan. Dear students, moving ahead, our next classification is based on mechanism of action of hormones, that is how these hormones act at their target tissues. This group is categorized into three types, that is endocrine, paracrine, and autocrine hormones. Let us discuss each of them in detail. First, the endocrine hormones. These are those hormones that are released into the blood circulation and carried to the target tissue all through the body to exercise their metabolic actions, are endocrine hormones. In the circulatory system, hormones bind to the carrier proteins and can exist in both, that is the bound form and unbound form. Just free or unbound hormones have biological activity. The binding affinity of a carrier protein to its hormone facilitates specific availability of the hormone in the circulation as well as at the target site. Once the hormone binds to its receptor at the target site by recognizing a unique location of the hormone, it permits the transmission of information carried by the hormone to generate a cellular response. Hormone receptors may be located either on the cell surface of a target tissue or intracellular, that is inside the target cell. Now let us discuss the second class, that is the paracrine hormones. Those hormones that are released into the extracellular spaces of the target cells and diffused to the neighboring cells. However, hormones exclusively having paracrine actions are locally secreted to act upon the surrounding cells. Paracrine factors are usually produced and secreted in much smaller quantities, showing specificity at the local target tissues and absence of the need to circulate all through the body. Dear students, let us discuss the third group, that is the autocrine hormones. These are the hormones that affect the same cell that release it and binding to the receptors on same cell surface. So simply, autocrine hormones target its own cell of origin in form of self-regulation. Dear students, we will be discussing one more classification of hormones based on the cellular receptors. Hormones are classified into two major groups, that is group 1 hormones and group 2 hormones. So group 1 hormones are lipophilic, that is lipid soluble in nature and are mostly derivatives of cholesterol. The exceptions are there, that is T3 and T4, because these are synthesized by attaching iodine to the amino acid tyrosine, and the benzene ring of tyrosine makes T3 and T4 lipophilic. Such hormones bind to intracellular receptors to form a receptor hormone complex via intracellular messengers through which their biochemical functions will be mediated. Examples are the estrogens, androgens, glucocorticoids, calcitriol, etc. Now let us discuss the group 2 hormones. These are hydrophilic, that is water soluble in nature, and include hormones such as insulin, follicle stimulating hormone, luteinizing hormone, gastrin, etc. Such hormones bind to the target cell surface receptor, where through the hormone act as first messenger and stimulate the release of certain molecules that act as the second messengers, which in turn execute a number of biochemical functions. Dear students, till now we have known the heterogeneous nature of hormones. Let's now briefly discuss the therapeutic uses of hormones. In general, hormones are employed pharmacologically for both their replacement and suppressive effects. Hormones may also be used for diagnostic stimulatory effects, for example, hypothalamic hormones to evoke target organ responses, or to diagnose endocrine hyperfunction by suppressing hormone hypersecretion. Example is the T3 from thyroid gland. Ablation of endocrine gland function due to genetic or acquired causes can be restored by hormone replacement therapy. In general, steroid and thyroid hormones are replaced orally, whereas peptide hormones like insulin and growth hormone require injection. Hormones can also be used to treat diseases associated with glandular hyperfunction, for example, acromegaly or hypersecretion in carcinoid syndrome. Estrogen receptor antagonists like tamoxifen are useful for some patients with breast cancer, and GnRH analogs may downregulate the gonadotropin axis and benefit patients with prostate cancer. Novel formulations of receptor-specific hormone ligands are now being clinically developed, resulting in more selective therapeutic targets. Modes of hormone injection like phenytoin may also determine therapeutic specificity and efficacy. Improved hormone delivery systems, including computerized mini pumps, intranasal sprays, pulmonary inhalers, depot intramuscular injections, and oral peptide formulations will also enhance patient compliance and improve ease of administration. Insulin delivered by inhalation has already been approved for use, and many others are under investigations. Dear students, cell-based therapies using the reprogramming of the human cells to perform differentiated functions either through differentiation of induced pluripotent stem cells or directed differentiation of one somatic cell type into another are under active investigation. Novel technologies offer promise of marked prolongation in the half-life of peptide hormones, thereby requiring infrequent administration. For example, once weekly preparation of exenatide, a glucagon-like peptide 1 analog currently used in the treatment of type 2 diabetes is currently undergoing clinical trials. So tremendous progress has been made in the therapeutic use of hormones. Although the delivery of insulin still requires frequent administration by injection and close monitoring by the patient, the purity of the insulin preparations has been vastly improved. thereby almost entirely eliminating the occurrence of uncomfortable local reactions at the injection site. Preparations with differing pharmacokinetics allow the normal physiology of insulin secretion to be more closely mimicked. Continuous administration via subcutaneous pump infusion greatly enhances therapeutic effectiveness in carefully selected patients. Closed loop systems in which the dose of insulin could be regularly adjusted depending on blood glucose concentrations are being actively studied. The implementation of such systems in the future would substantially reduce the burden of this disease. Hormones are biologically powerful molecules that exert therapeutic benefit and effectively replace pathologic deficits. They should not be prescribed without clear-cut indications and should not be administered without careful evaluation by an appropriately qualified medical practitioner. My dear students, this was all about today's lecture. Hope you have understood it well. Thank you. Goodbye.